welcome to my channel. Let's start a journey with Neera. Today we're going to learn how to do homology modeling. Just imagine you have an amino acid sequence, but you don't have any protein. That means you know what is the protein, but what if you don't know the 3D structure of the protein? Here only the protein sequence is available here, but there is no 3D structure of the protein is available. So how can we determine the 3D structure of the protein? Yes, we can determine it through two processes. Here one is the experimental method. What is this method? We use this method to do the modeling and we may get a 3D structure of a protein. There are two methods. One is the X-ray crystallography and another is the NMR. Okay, through this method we may determine the tertiary structure of the protein, but here is a disadvantage here, that is this method is so costly and time consuming. You may need much time to determine the protein because that's so time consuming. So what can I do? Yes, we have another one method, that is computational or in silico method. Yes, obviously that is so easy, not time consuming and less costly. Of course we will select this method because it's so beneficial and advantage, so many advantages available here. So what can we do? Actually we have a BLAST, that means a protein sequence, and we select the highly identical sequence from here, and then we may predict the based on the highly identical sequence we may predict a structure. So what are the steps? How can we determine this tertiary structure of the protein through this computational in silico modeling? Here are some steps. If we follow these steps, maybe we may complete the homology modeling of protein. Here first is template selection. That means you already have a BLAST, but you don't have a model, so you need a highly identical sequence, and that means you may collect this highly identical sequence, that is called the template. Then you need to do the modeling, and so you have a sequence alignment. That means you have a template, that's also called sequence alignment, and now you have to build a model. If you know the template and if you go through this process, yes, we may get the model which we need. Then the another one is the loop modeling and side chain refinement, and then the model refinement using the energy function. What is this refinement? That is actually whatever a model and what I have built. Okay, through all of this process you can get a tertiary model using online server. Because you have a question, how can we do all of these steps, where we may do? Yes, we have a online server that is called Swiss model. Through this online server we will predict the 3D or tertiary structure of the protein, and in my next video we of course perform on this Swiss model online server. We will do the practical of the homology modeling. So let's see the next video, how can we predict tertiary structure of the protein. Till then, thank you very much.